The bone marrow provides an environment where various cellular components of our blood develop and mature in a process called hematopoiesis. Hematopoiesis starts to occur when stem cells start to change and acquire different properties as they differentiate. Such stem cells can either differentiate into common lymphoid precursor cells, which can give rise to T and B cells, or common myeloid precursor cells, which can give rise to almost all the other cellular components of the blood. As these stem cells differentiate into common myeloid precursor cells, they start to express the transmembrane protein CD33 on their surface. Cells of the myeloid lineage are known to be involved in the development of acute myeloid leukemia, or AML. Furthermore, these malignant cells are known to express CD33 on their surface. Leukemic cells in AML rapidly proliferate within the bone marrow, interfering with the normal process of hematopoiesis. AML is primarily treated with chemotherapy. However, scientific research strategies involve developing antibodies that specifically bind to CD33. As CD33 is expressed on the surface of AML blasts, it represents an attractive therapeutic target. The interaction between the antibody and the CD33 antigen can trigger antibody-dependent cellular cytotoxicity. Boehringer Ingelheim is currently developing a novel CD33 binding antibody, which has been engineered with the goal to deliver antibody-dependent cellular cytotoxicity. Boehringer Ingelheim's CD33 antibody is designed to recognize and bind to a specific epitope on CD33. BI 836858 intends to cause decelerated internalization, which may lead to a prolonged exposure on the cell surface. It also has a modified Fc region, which intends to enable it to bind with high affinity to the Fc gamma receptor 3A found on the surface of immune effector cells. Preclinical studies have shown that this engineered antibody exhibits antibody-dependent cellular cytotoxicity. In addition, it has been shown to display antibody-dependent cellular cytotoxicity against AML blasts in vitro.
Pertuzumab is a HER2-directed, humanized monoclonal antibody. It is the first in a new class of agents that target HER2, called HER2 dimerization inhibitors, or HDIs. HDIs bind to the HER2 receptor to block dimerization of HER2 with other HER family members and suppress HER2-mediated cell survival and proliferation. Pertuzumab, a HER2 dimerization inhibitor, is designed to bind to domain 2 of HER2 to block ligand-activated pairing of HER2 with other HER family members, including HER3. Preclinical studies demonstrate that inhibiting HER2-HER3 dimers interrupts both the MAPK cell proliferation pathway and the PI3K cell survival pathway, and ultimately tumour growth. The pertuzumab mechanism of action is complementary to that of trastuzumab, another anti-HER2 monoclonal antibody, which has already been established as an effective treatment for HER2-positive breast cancer. Preclinical studies have demonstrated that combining the anti-HER2 activity of trastuzumab with the HER2 dimerization inhibition of pertuzumab offers a more comprehensive blockade of HER2-driven signaling pathways than either agent alone, resulting in synergistic anti-tumor activity.
Anti-HER2 mechanisms of approved HER2 inhibitors. The development of many breast cancers are driven by the presence of HER2. HER2 is a transmembrane tyrosine kinase receptor which belongs to the family of the EGFR, epidermal growth factor receptor. Its unique feature, which differentiates it from the other members of the family, is the absence of a known ligand. HER2 is overexpressed in 25 to 30% of human breast cancers and has been determined to be an adverse prognostic factor. Since the level of HER2 in human cancer cells with membrane overexpression is much higher than in normal adult tissue, there is potentially more sensitivity to the toxicity of HER2 targeting drugs. HER2 overexpression is typically found in both the primary tumor and in metastatic sites, which provides the rationale for the effectiveness of anti-HER2 therapy at all disease sites. Trastuzumab. Research has therefore focused heavily on HER2 inhibitors as anti-cancer agents. Trastuzumab is the first of such agents which was registered for use in patients with HER2 overexpressing breast cancer. Trastuzumab is a recombinant monoclonal antibody directed against the extracellular domain of the tyrosine kinase receptor HER2. Trastuzumab has shown clinical activity in HER2 overexpressing breast cancers, and at present is currently approved for patients whose tumors have this abnormality in both the metastatic and the adjuvant setting. A number of different mechanisms of action has been attributed to its anti-HER2 activity, including antibody-dependent cell-mediated cytotoxicity, prevention of HER2 truncated membrane-bound fragment following HER2 overexpression, and HER2 receptor downregulation, though these are still subject of discussion. However, binding of trastuzumab to the HER2 receptor and subsequent inhibition of HER2-mediated signaling transduction is thought to be the main mechanism of action of trastuzumab. Pertuzumab. Pertuzumab is a novel, fully humanized monoclonal antibody which binds to domain 2 of HER2, a portion of the extracellular domain essential for dimerization, while trastuzumab is known to bind to domain 4. This binding of pertuzumab efficiently sterically blocks ligand-induced homodimerization and, importantly, HER2-HER3 heterodimerization, which activates downstream survival signaling through pathways such as phosphatidyl inositol 3 kinase PI3K-AKT, whereas trastuzumab has only a minor effect in the presence of a ligand. Pertuzumab was approved in mid-2012 for use in combination with trastuzumab and docetaxel to treat patients with metastatic or locally recurrent unresectable HER2-positive breast cancer who have not received previous anti-HER2 therapy or chemotherapy for their metastatic disease. TDM1. To increase the potency of antibody-directed therapy, the specificity of the antigen-binding site has been combined with a wide variety of effector agents, including toxins. This led to the development of the antibody-drug conjugate, ADC, trastuzumab emtansine, TDM1, which is comprised of the anti-HER2 antibody trastuzumab bound to the potent antimicrotubule cytotoxic agent maytansine, DM1, by a thioether linker. TDM1 uses trastuzumab to specifically localize the highly active chemotherapy to HER2-positive tumor cells. The trastuzumab portion of the ADC selectively binds to HER2 receptors and is internalized by the cancer cell. In addition to the anti-tumor activity initiated by trastuzumab, once internalized by the cancer cell, trastuzumab and the linker are degraded through lysosomal degradation, and DM1 is selectively liberated, causing cell cycle arrest and apoptosis through direct inhibition of microtubule assembly and polymerization. The linker has been specially designed to prevent DM1 from detaching from trastuzumab prematurely. This may lead to a reduction in the size of the tumor. As trastuzumab resistance remains a challenge to the management of advanced breast cancer, studies with TDM1 may offer an additional option for these patients. Responses to TDM1 in recent clinical investigations have been the basis of the recent FDA approval for TDM1 for the treatment of patients with HER2-positive metastatic breast cancer.